So before I starting to share my presentation, I just wanted to thank the KAT6A Foundation to having us and giving this small presentation that is part of a joint effort between my lab at the Center for Regenerative Medicine and the KAT6A Foundation to create a patient-derived iPSC bank. And I was supposed to be there, so I'm sorry I'm missing this wonderful event and the opportunity to get in touch with everybody. And this was because sadly a flight got canceled. And I'm mentioning this because I would like to highlight the wonderful efforts from Jackie, Natasha, Carly, and Bawika that since we were facing this challenge of me not being there to do this sample collection, they really stood up to the challenge and went above and beyond to help me get these samples for this bank. So a very special thank you for everybody who's supporting this effort. Okay, having said that, let's now start presenting, sharing this. Okay. So the goal of this presentation, let me know if at some point you cannot hear, please. The goal of this presentation is to basically share with you this huge effort that we are strongly convinced is actually going to help adopt the KAT6A and KAT6B community, the patient community, and of course through sparking research. All these forum is just to go over a few concepts, but then I would love you to make the most of this opportunity to ask all the questions that you might have. And if you're considering participating in the study, please don't hesitate asking any question. I'm going to make sure to answer all of your questions in the best of my abilities. So basically what I want to share with you is what are these iPSC? Why are they so important currently in science and during research endeavors? What is an iPSC bank? What do we actually call a bank, and what's the impact of that of that reservoir of cells in terms of how will that help the community, the patient community, and how will that help to trigger a new research collaborations? So let's start first by what are these iPSC, these iPS cells. So basically what we consider iPSC is one specific type of stem cells. Stem cells have the possibility to generate any other cell of the human body. But in this case, these induced pluripotent stem cells come from somatic cells. That means they can come from a skin biopsy, or they can be generated through a blood draw, which is what we are doing now at the conference on site. And what the amazing power of this is that we can take those bloods from a patient with all the genetic background of that patient, and then use those already differentiated cells that have already very specific purpose and context. We can reprogram them, that's the term that we use, to make them go back into an embryo-like state. So they will again have this ability to generate any other cell of the human body. And this in concept is very powerful, but currently we are strongly working on making the best protocols possible, so all the tissues and cell types that we generate in a dish can really represent what we have in the human body. So we are working towards making those protocols to generate these different cell types, like neuronal progenitors or cardiomyocytes, or different cells of the nervous system. So we can use those to actually do the research that the community needs. So that's basically the strength of these cells. They are cells that have embryo-like characteristics. They can generate any cell of the body, but we take them from the specific patient. So then all the therapies or all the things that we can think in the future, in the long term, have the advantage that if we want to think of a cell therapy where we have this cell that is corrected or that has undergone some kind of engineering process, we can use those cells to actually take them back into a patient, and the patient and this cell type won't have any kind of immune response because they are from the same individual. So that's one of the biggest advantages. The other advantage is that these are an endless source of of cells. Because one of the biggest challenges that we have is how limited specific cell types are. But now we have these cells that can reproduce, that we can kind of duplicate and replicate in the lab, and then differentiate it in different cell types. So that means that we will have a source that theoretically has the possibility to be kept for very, very, very long periods of time. So that's basically the main concept of what these cells are. But now you might be asking, so why do we need to have these particular cells when we have so many other different models? Are these cells the perfect model? And so then why are we investing also in research in mice or in fish and in different cell types? And the truth is that the whole scientific community is working towards achieving an ideal model, a model that will best represent a patient, and that particular patient, not just any patient. So here, I really like this cartoon that was made by one of my collaborators in Utah, where they basically try to show where we are in the development stage with each system model that we are using. So here we have in one of the axes the phenotypic relevance, how what are the clinical manifestations that we can best reproduce with these models. In the x-axis, we have the human and patient relevance, the genetics, the backgrounds, how well that model will represent actual human biology. And then, are we going to be able to do to ask biologically important questions in those models? Are we going to be able to determine and identify the cellular molecular cause that is then giving us the clinical manifestations of these patients? So if we take into consideration all these three main areas, we can see how different models are a bit ahead or behind in order to get to the ideal, what we consider the ideal model. And as you can see here, I'm super happy to see zebrafish because I am a zebrafish researcher too, not only a brain organoid researcher next to model. And you can see how each of these different models have their own specific strengths and weaknesses. And the answer to what's the best idea or the ideal model nowadays is a combination of every single model that we can develop to ask specific questions. And I wanted to share this with you because I know that in the community, you will see research endeavors that are not only done for IPSC Bank or for making a mouse model or a zebrafish model. Every single thing is super important, and all of them will contribute to help us better understand the disorder itself, and then how can we, after gaining that body of knowledge, how can we apply that into improving the health of our children? So that's basically the goal of this cartoon. So, okay, we know what are these iPSC cell, and now I'm saying we want to make a bank. So the Center for Regenerative Medicine is a unique center because we have a concept of open source biology. That means that every protocol, every research endeavor, every cell or every tool that we develop in this center has to be shared with the whole community. We won't charge for it for our own benefit, and we won't wait or hold this sharing because the data is not published. So everything that we generate, we share. For that reason, creating an iPSC bank in our center is so important. And these endeavors of sharing are done between different institutions. For example, I want to start a collaboration with Dr. Harris to share this cell. So these things are already happening in the community. What we want to do here is to formalize it and building a cell line that can cost $10,000. Now we can just ship that cell to a lab by just shipping cost or basic maintenance cost as low as $600, which is huge for some research labs that, for example, I can put myself into that bag, that we're struggling to get funds and to actually improve research. So this kind of video is just to show how you can find this online. If you put CREM Center for Regenerative Medicine and you go to the catalog, you will be able to the biobank, you will be able to find this bank. So the bank is basically, just to put it in like terms of, okay, yeah, you're talking about a bank, but is that like a freezer? Is that like a drawer? Is like a different building? What is that? So basically what we do in our lab is we we gather the samples from patients, the blood. We isolate cells that will allow us, from that blood sample, that will allow us to generate these induced pluripotent stem cells. We store it with medical records from those patients, and we store it in special freezers that will help us to maintain or to preserve the quality of those cells for a very, very, very long time. And then we create a database online that is available for everybody to look at, like the one that I'm showing here in this presentation, where you can actually search for a specific disorder. Right now, for example, my research program is Kabuki syndrome, which is another rare disorder. So you can find that, and how I'm starting to build that database. And now, thanks to the efforts of the KAT6A Foundation and my lab, we're gonna include this, and with your help and support doing this, we're going to have KAT6A and KAT6B disorders in this database. So I think that it's very important that you know that this is the kind of effort that I call domino effort. You just move one piece, and then suddenly many other pieces are following that specific movement. So creating this bank will help us to support research endeavors all around the world virtually, because we do have some barriers for shipping biological material through borders, but those are things that we can work on, but we can share the cells to whoever requests them. And this is not only with research labs, but also with industry. So if for some of you that are already consented, you probably were consented knowing that this material that we are going to generate from the patients will be shared to spark research and collaborations with academic institutions and with industry. One important thing that I do want to mention here, that I know has been some of the questions that have been asked to our research coordinator, is how are we protected? How I don't want my child information all out there. So it's very important that you know that our main concern is to protect you and your children. So that's like the main goal of all this study. So what we do is, after we generate the draw, the cell from the blood draw, and we keep all your medical records, we randomize the samples. We put a number or an identifier that cannot, that's the number that is going to be shared with other institutions, not your name, not your personal information. And even though we might share your medical records and the cells, all the identifier information will be removed, and only you will have like the ID, unique ID. And there is only one specific group of people at Boston University who will be able to correlate the sample with the person who donated that sample. So that's super important for me, that you know that our main goal is to protect you and to protect your confidentiality. So just as a summary, and just to kind of have the big picture of all of this, and how can this help us? So sadly, we're at the stage that all our research endeavors are not enough to help the current generation of patients. So we're working super hard to at least understand more about the cellular molecular basis of KAT6A and KAT6B disorder, with the goal of, in the future, and we don't know what that future is in terms of time, that we can actually help a future generation of patients. But the way this work is that we want to achieve collecting blood from genetically diverse patient population. And in our case, we're doing it from blood, but it can also be a skin biopsy. And then we separate these cells that have genetic material. In this case, we call them PBMCs, that stand for peripheral blood mononuclear cells. We use a specific research strategy to force those cells to go to an embryo-like state. And that's when we have our induced pluripotent stem cells. And, oops, sorry. I have all the Zoom windows here too. So then we store all those cells in our bank, and we share them worldwide, with the hope that the more research is done with these samples in different tissue, and the more labs are interested, the more industry is interested, we are gonna fast forward a little bit towards finding implementing functional testing and drug screening that are specifically done in these patient-derived samples. So that's kind of the big picture of this. And just I've shared this with you before. I just want to take the next 3 minutes to show an example that is particular of my research program, where you can have these samples from patients, like the effort that we are doing now at the conference, but we can also engineer this iPSC line through CRISPR and Cas9 mutations. We have these iPSC lines that, again, potentially can generate any part of any cell type of the body. In the case of my lab, what we do is to induce neuronal tissue. And you can see here these rosettes are a specific cell type that has that is neuronal, but is a progenitor neuron that can then differentiate in different neuronal types. And then we use these to go a step further and then generate what we call brain organoids, which is basically a three-dimensional representation of neuronal tissues that allow us to better understand the interaction between different cell types. And we can also do two-dimensional cell cultures to ask different biological questions. And we use all these models, in my case, together with zebra-fish disease modeling, to understand more the cellular and molecular elements that are contributing to the clinical manifestations of the patients. So this is just an example. This is what a brain organoid looks like. This is at day 1 when we generate these specific tissues. At day 7, you can see how this structure here that we call rosette, that has a lumen inside, that would be like a resemblance of what is a brain lumen, like a brain ventricle. And then at day 14, this increase of size, and then we have differentiations of different neurons around that lumen. So here at the top, this is just to show you how this works when we have another line that actually has a pathogenic variant of the gene that we're studying. In this case, is KMT2D, that is causative of Kabuki syndrome, this other rare disorder. And you can see here at the top the size and the morphology of our non-disease scenario. And here, these two other row of pictures are showing you what it is a disease scenario. And you can immediately observe that the size is different and the morphology is different. And we quantify this to better understand the differences between disease versus non-disease scenario in a quantitative way. And then this is very overwhelming, a very overwhelming picture, but I just want you to take 2 take-home messages from this. The first one is that we have the ability to stain different cell types. In this case, this is a very thin slide of one of those brain organoids, and then we stain specific cell types, and we took them to a super resolution microscope. And in green, we can identify those neuronal progenitors that I mentioned that have the ability to then differentiate into different neurons. And here in magenta, what you observe are those neurons that have already differentiated. So we have non-differentiating neurons in green and more differentiated neurons in magenta. And in this case, WTC is a non-disease scenario, and here KMT2D is the disease scenario. And you can already observe that we do not have all that magenta lines in between these green cells, like you can observe in the non-disease picture. And then all these nucleus, all these green spots that you see here have a different morphology. And we then take that information to ask more biological questions and to understand more. And one of the things that we do is what we call single cell RNA-seq. And I do want to spend like 1 minute explaining this, because it's something that that is like fashionable right now, if I can say it some way. Every lab is aiming to do this. It's a technique that allow us to see and to read all the transcriptome that every single cell of a specific tissue or organ is having. So a good way to understand the differences between a cell that has the disorder and a cell who doesn't have it, that doesn't have it, is by doing this kind of analysis. And actually, this is what Carly Golden, who's there with you, is learning how to do in my lab with KMT2D samples. So here in this map, this is like a way to squish loads and loads of information in 2 dimensions, and every single spot is one cell of those brain organoids. And you can see in one color here, in these reddish colors, we have the non-disease sample, and then in this cyan bluish is the disease sample. And you can immediately see how KMT2D samples are missing some specific cell population. So now it's amazing to be able to ask, okay, what are those cells? And what are the pathway or the functions of those cells in the organism? And we can use that to then start designing experiments to use for drug screenings, or to even understanding a bit better what the clinical manifestations of the disorders. And with that, I would like to thank you. I would like to leave like at least, and we have like, sorry, 2 or 3 minutes for a few questions. This is my email. You please feel free to write an email to me if you have any question. Carly is there, and she also has the contact information of our research coordinator in case you now decide that you would like to participate in the study, and we can consent you by phone and you can donate your sample. So if you have any question, I would love to be able to answer it.
Hey, Angie, this is Jordan.
Hi, Jordan.
How are you? Okay, it was great. For sample contribution to the IPSC Bank, I mean, so for a family that contributes samples, would you expect or need that family to contribute multiple samples over time, or is one sample enough? And then second, how many samples would you need from the KAT6A or KAT6B community writ large? You need 5, 10, 100, like is more better? Or is there once you get enough, as long as those cells persist, do you need to continue collecting more and more and more and more over time?
That's a wonderful question that I actually forgot to address. So thank you, Jordan, for asking that question. Actually, we just need this sample given once, because unless, and this is something that I just want to be very clear about, we draw the sample, and since we're in a different state and we are doing all this logistic in order to have them shipped here, and immediately here I can process those samples, the only thing that can happen is that the viability of that sample can go down a little bit, but we are going to try to avoid that at all cost. But once we have that sample, we can store that's the same sample that we use time after time. We won't need more samples from patients. And that's one of the goals of this effort. We want to generate a source so children are not being poked all the time. So once we have this, then we share it. We share the iPSC. So every research study that will need iPSC development, then they won't have to have your kids poked and drawn blood. So it's just once, unless something happens from now until I get those tubes in my hand and those cells isolated from there. So ideally it's just once, and that sample just stays there in that database.
Can I say this? Hi, Angie, it's Carrie Ginsberg. Can we ship cells that are stored? We have cells stored at CHOP. Can we ship them to your lab, or do you?
Yes. So let's say there are families that are not present there, or now we cannot consent them for a specific issue. Ideally, the viability of the cells that we need are good. Ideally 2 hours, but we can push it up to 8 hours. So yes, we can coordinate with the lab in your home state and see if we can work out a way that they can spin them down. We would send the tubes, we would cover the shipping costs, and we would do everything we can to get that sample shipped to our lab. As soon as we receive it, we do the isolation, we check cell viability, and then we say, yes, this sample is good. So for doing that, what you need to do is contact Natasha or myself and say, I would like to do this. We communicate with you, we have you consented, and we start analyzing the logistic of what's the best way to get those samples to us. And it's not only the patient sample. I also want to highlight that we are taking sex-matched parents. And the idea is because that we want to make our best effort to have all the research tools and research abilities to have a good control to compare. with a disease scenario and a non-disease scenario. So the genetic background of the parents is also very important in this effort. That's why we are requesting the parents to donate sample too. But if that's not possible, just the patient sample is enough.
Thank you.
Okay,
When
if you do
a
have
clinical trial requests samples from the stored bank, are you making a profit off of giving those samples to that researcher?
Yes, thank you for that question. No, we do not make any profit at all in the consent form. It's a very wide consent, and we work very hard to make a consent that will allow us to better share these samples. So we say that we state specifically, and we are legally bound, that we are not gonna make any profit by sharing these cells. What we charge are just basic maintenance of the bank, which is very, very minimal, and a shipping cost. That's the only thing that we ship to institution. But if institution says to me, because I did work in Argentina in a lab and we did not have money for anything. So if anybody comes to me and say, I really cannot even pay the $600, just talk to me. I mean, I will do everything we can to make research happen. Okay. So if there are no more questions now.
Yes.
And,
Dr.
yes.
Serrano, how are you? I'm Eduardo Kabuki. Don't know if you remember me. We were trying to get in touch by email a couple of weeks ago. Yeah, we're the family from Colombia. We are very interested in kind of like taking the samples, but our little boy, we couldn't bring him because of special issues. But is there any possibility that we can do that from over there? So
Yes.
do we send the samples from Colombia or something special? Or does it have to be here in the United States?
Yeah, thank you so much for that question. I apologize for the logistic problems in communicating together. So the problem is that to bring in biological samples to the United States, it's another world of problems. So what we need to do is to work with a research institution there in Colombia or with a hospital and see if they've already had experience to bring samples in. The borders, especially in the United States, that laws are very strict, is very, very, very hard to bring biological samples. I'm not saying it's impossible. So what we need to do is to see some logistics, and we need to talk about this. I apologize, we haven't done this yet, but what we need to do is to first identify a research institution there who might be willing to support us in this. And if that doesn't go through, if that's not possible, then I need to start assessing different mechanisms. It's a big challenge, I won't lie, and I'm really sorry that some political and border laws are making this difficult, but we will assess that. And I'm not aware, because the other thing, yeah, I'm not aware of a lab that's doing this. It's easier to ship iPSC than blood. So if there is a lab already doing this in Latin America, we need to think about it. I'm gonna see if in Argentina there is anybody doing it. So there is a lot of thinking and strategy, creative thinking to do here. But I'm not saying it's not impossible. We just need to work a bit harder for it.
Okay, thank you, Angie.
Thank you, everybody. Please feel free to contact me, and I'm gonna be with my phone next to me. If anybody has a specific question, Carly has my number, Bowie, Natasha, so please feel free to contact me. Thank you, and I hope you continue enjoying this amazing set of talks that you're about to hear. Thank you.